Ernest Mayr, the author of Animal Species and Evolution, discovered in his fieldwork new knowledge of species, including 26 birds and 38 orchids. Charles Darwin, the author of Origin of Species, figured out how a single species can change over time and why. But how species, in general, can split into two was not part of the concept. The takeaway is that we need to do more than just explain how traits arise, but also how species arise. The origin of species is also known as speciation. A species is a discrete cluster of animals, and the understanding of how these species arise is called speciation. In the book, it is defined as a group of individuals that resemble one another more than they resemble members of other groups. Why is this not an effective definition? Males and females from one species are confused with females and males from other species. Variation also plays a big role between interbreeding groups. Different species look either exactly alike or nearly alike can be called cryptic species. Fruit flies have 9 different species, one of them being Drosophila. The reason that they can have 9 different species could be that they reproduce in isolation. The prime example are humans. Because there are no main differences, the definition would not be able to categorize into species. The question is, at what point are differences and similarities among populations large enough to classify species? Science draws the line at sexual reproduction. The reason we classify distinct species between phenotypically similar populations is their reproductive isolation. Reproductive isolation happens when species only breed and reproduce within their own gene pool. While they can coexist in some locations, they do not exchange genes because there are barriers. An example can be 2 species of monkey flower that are pollinated differently, one by a hummingbird and the other by bumblebees. The biological species concept describes a species as a reproductive and an evolutionary community. Yet sometimes this isn't foolproof, considering that there are those that do not sexually reproduce. Under the BSC, if you can explain how reproductive barriers evolve, you've explained the origin of species. Species that are each other's closest relatives were often separated in nature by geographical barriers. Mayer argues that the reproductive barriers are also a byproduct of natural or sexual selection that cause geographically isolated populations to evolve in different directions.
Suppose, for example, that an ancestral species of flowering plant was split into 2 portions by a geographic barrier. Like a mountain range, one population lives in a place having a lot of hummingbirds but only a few bees. In this area, flowers will evolve to attract hummingbirds as pollinators, so the flowers in this area would become red, have deep tubes with nectar. The population on the other side will have the opposite, few hummingbirds and a lot of bees, which would mean that the flowers would attract bees more as pollinators, so the flowers would become pink and evolve shallow nectar tubes with less nectar. Divergent selection is a selection that drives different populations in different evolutionary directions. 2 populations' mating preferences would prevent them from mixing genes, so this would mean that they would be considered 2 different species. The study of speciation tells us that species are evolutionary accidents. The clusters don't evolve to provide balanced ecosystems. They are simply the result of genetic barriers that arise when spatially isolated populations evolve in different directions. Biological speciation is when populations change genetically to the extent that their members no longer recognize each other as mates, or their genes can cooperate to produce a fertile individual. The theory of geographic speciation states that the evolution of genetic isolation between 2 populations requires that they first be geographically isolated. On the other hand, the theory of population genetics is the splitting of a single population into 2 genetically isolated parts, which is very difficult if they retain the opportunity to interbreed. The Drosophila or the fruit fly experiment. Alan Orr and the author Jerry Coyne measured three types of reproductive barriers in the laboratory: mating discrimination between the pairs, and the sterility and inviolability of their hybrids. They predicted that the reproductive isolation between species increased steadily with time. Genetic barriers between groups became strong enough to completely prevent interbreeding after about 2.7 million years of divergence. In conclusion, for fruit flies, the origin of new species is a slow process.
Origin from the stars. The discovery of speciation has very much followed in the footsteps of the discovery of a star's own evolution. Both processes, while beyond the human lifetime scale, are pieced together through snapshots of evidence, which, when put in a continuum, give us a better understanding of the process. Various stages of star evolution have been seen which, when pieced together, allow us to understand their evolution and extrapolate from what is known through this continuum. The process of speciation has very much followed this. Speciation. The process of speciation has further been pieced together through evidence of species isolated by geographic barriers such as mountains and rivers, as well as by species descended from a common ancestor. A general process of speciation can be as follows. An initial population of species becomes isolated and split due to a geographic barrier. Over a long period of time, these two populations are stuck in reproductive isolation. Over many generations, this reproductive isolation leads to the rise of new species from which reproductive barriers develop through evolution. This process is often argued against because through our own human lifetime, we do not see species arise. But this process is always at play, albeit at an extremely slow rate. Geographic barriers. Geographic barriers are what biologists attribute to be the most common propagator of speciation. Darwin himself accounted of new species of flowers arising in the same small area. This has led biologists to believe that speciation can occur without geographic barriers. A typical scenario in which geographic barriers are at play can be seen on the diagram on the right. An original beetle population, split in half through a river, has effectively split the population in two. After many generations of reproductive isolation, both populations evolve to the state of not being able to merge with the other population. Sympatric speciation. Sympatric speciation is the hard split from one population into two species within the same area. This type of speciation occurs very rarely, as there would be a tendency for diverging forms of the population to interbreed and return to an equilibrium species. While this seems unlikely, evidence has shown sympatric speciation to be possible only, however, under restrictive conditions uncommon in nature. An example of sympatric speciation can be seen on the diagram on the right. A small population of two species of flowers in a single area eventually interbreeds to produce a new species. This new species, through asexual reproduction, populates in the new area, and soon three species are within the same area. Sympatric speciation continued. While evidence for geographic speciation is relatively easy to find, evidence for sympatric speciation is not. Two related species living in one area doesn't necessarily show sympatric speciation because there is the possibility that over time, geographic barriers have reduced, allowing both species to come into contact. One can tell if species arose in the same place by finding isolated patches of terrain that are generally too small to have geographic barriers and look for closely related species. If there are, one can assume, due to low probability of geographic isolation, that sympatric speciation was at work. Examples include the cichlid fish in two tiny lakes in Cameroon and the kentia and curly palm trees on Lord Howe, an oceanic island off the coast of Australia. On the diagram on the right, one can see the difference between allopatric and sympatric speciation. Allopatric speciation involves geographic barriers separating the population into two, whereas sympatric speciation does not. We can also see in the diagrams on the right variations of the cichlid fish, as well as an example of the kentia palm tree. Two types of sympatric speciation. Two special forms of sympatric speciation are evidence of speciation in action. Allopolyploid speciation occurs when two different species that live in the same area hybridize and produce a sterile hybrid due to different numbers or types of chromosomes. This new sterile species would be unable to mate with similar hybrid species or the parental species unless its chromosomes were doubled, which would then provide for a fertile hybrid that would give rise to new species. An example of this can be seen in the diagram. One species of normal gametes and a second species with polyploid gametes produce a hybrid polyploid gamete, which then goes on to produce a hybrid species. Two types of sympatric speciation continued. Autopolyploidy is the process by which a species arises by simply doubling all of the chromosomes in the new species. Autopolyploid hybrids are fertile and can reproduce with other hybrids to produce fertile hybrids, but produce sterile hybrids when mating with the original parental species. In the diagram below, we can see the results of autopolyploidy. The diploid parent of 2N chromosomes gives the offspring two sets of chromosomes, making it a polyploid of 4N. Other examples include the triploid, which are offsprings that contain 3N chromosomes and are usually infertile. The tetraploid are offsprings that are fertile but contain double the original chromosomes. Polyploid speciation. Both polyploid speciation processes require a rare event in two successive generations: the formation and union of sperm and eggs with abnormally high numbers of chromosomes result in a hybrid species. This speciation seems rare, as it is in animals, but it is very common in plants. Because of a plant's ability to produce millions of eggs and pollen, polyploidy has higher probability, as is evidenced in the 1/4 of flowering plants that are polyploid. One very common plant that is polyploid is the banana. In the figure on the right, we can see that there are two bananas. The original parental banana is on the right-hand side. It contains seeds, whereas the hybrid banana on the left do not contain any at all. When we usually see black specks on a banana, they are actually remnants of aborted plant ovules that don't turn into seeds because the hybridized chromosome sets abort the ovules. Polyploid speciation continued. Polyploid speciation in animals is much less common. Most polyploid animals arise from asexual processes, but the red viscacha rat of Argentina is the only known sexually reproducing polyploid mammal. The reason that there are less frequent numbers of polyploid animals is not known, but we do know that plants are more commonly polyploid because of their ability to self-fertilize and produce related species. Below we can see an example of the red viscacha rat, which contains 112 chromosomes, the most ever seen in any mammal. The power of polyploid speciation. Polyploid speciation is different because it involves the change in chromosome number instead of the changes in the genes. It can also be a much faster process, only requiring 2 generations for a new species to arise. This fast process of speciation gives further credence and satisfaction to the process of speciation and allows us to see it happen within our lifetimes. One example of this process in action is the Welsh groundsel. The Welsh groundsel, which can be seen on the right, has been studied and found to have been produced from the common groundsel and the Oxford ragwort. Both parental species were taken into a lab and hybridized and proved that they both hybridized to form the Welsh groundsel. This process of checking parental hybridization has allowed us to see polyploid speciation in action and to thoroughly predict what we would expect. Seeing how far we've come. Polyploid speciation has provided for significant evidence in the theory of evolution. By seeing splitting lineages in the fossil record, related species separated by geographic barriers, or polyploid speciation occur before our very eyes, the mystery of speciation in the context of evolution has come a long way since Darwin and continues to have a long way to go. This concludes our presentation on Chapter 7, The Origin of Species.